So we'll present 4 studies. 2 are actively open, actively treating patients, and then 2 are going to be activated, are in the process of activation. And those are the ones that we will highlight. So the first one that we would like to present is Dr. Mark Dixon talking about SARC-41.
Good morning, everybody. So I won't belabor the background too much because you've all heard about this study before. But in brief, there are 3 commercially available CDK4 inhibitors, abemaciclib, ribociclib, and palbociclib. All have been evaluated in single-arm phase 2 studies in liposarcoma. Abem is arguably the best in class with a response rate in phase 2 of 17% compared, for example, to about 4% for palbo. So SARC-41 is a phase 3 investigator-initiated randomized double-blind trial with a primary endpoint to determine the progression-free survival in patients with abemaciclib versus placebo and standard secondary endpoints. And patients have the opportunity to cross over if they progress on placebo. Eligibility key points here were metastatic or recurrent differentiated liposarcoma. Patients with purely well-diff disease were excluded. They had to have progression of disease by RECIST 1.1 in the 6 months prior to going on study. Any number of systemic therapies were allowed, including no prior therapy. And this is the treatment plan. So patients were randomized 1-to-1 to abemaciclib at a standard dose of 200 milligrams PO twice a day versus placebo. Scans every 6 weeks, and then crossover at progression if they were unblinded and found to be on placebo. So the statistical design, 108 evaluable patients, 54 in each arm, with PFS as the primary endpoint, and it's powered to detect a hazard ratio of 0.6. So this study was opened at 9 SARC cancer centers around the United States, and they are listed here, and I thank them all for contributing to the study. This opened in the shadow of the pandemic, with the first patients going on in the fall of 2021, and enrollment was complete in the fall of 2024. And as of now, many patients are continuing on study, and the final analysis will occur when 95 progression events have occurred, so we have not yet reached that. So I hope at some point, the next time you hear about this study, I'll be able to actually give you a result. So thanks very much.
The next study will be done by Candice Haddock, SARC-44.
Good morning, everyone. It's great to see you all in person today. I'm happy to give an overview of SARC-44 and discuss where we are with enrollment. So we all know that tyrosine kinase inhibitors have really revolutionized the management of patients with GIST and have improved outcomes, but almost invariably resistance does emerge on treatment, and that's through secondary mutations in the KIT gene, specifically in the ATP binding pocket or in the activation loop. So bezuclastinib is a selective type 1 KIT inhibitor, and it was designed to bind and inhibit KIT with AL mutations. And this complements the activity of sunitinib, which inhibits KIT with AP mutations. There is an ongoing phase 3 PEAK trial, which will evaluate this combination in patients in second line, and that will be compared to sunitinib. But in SARC-44, we're looking at this combination in patients that have already progressed on sunitinib with a primary endpoint of median progression-free survival. We're also hoping to discover mechanisms of treatment response and resistance. So patients will go through a screening period with a TKI washout. We hope to enroll 40 patients and have 35 evaluable for the primary endpoint. Patients will start with bezuclastinib alone for 2 weeks and then add sunitinib and then continue until progression or unacceptable toxicity. We do have collection of ctDNA at baseline after the washout, after 2 weeks on bezuclastinib alone, after 2 weeks on the combination, at their first response assessment, and then at the time of end of treatment. This will allow us to ask really interesting questions about how resistance patterns are emerging or falling on treatment. Out of the 40 patients, 20 will also participate in a PET-CT biopsy cohort. So after washout, patients will have a PET scan. They'll have a second PET scan after bezuclastinib alone and a third PET scan after the combination. And with this series of PET scans, we're going to select a lesion to biopsy and prioritize one that's resistant to the combination. We can then use that tissue to ask questions about mechanisms of resistance and really dive into that biology, looking at targeted exome sequencing, KIT activation, allele-specific PCR to know if the KIT mutations are on the same allele or in different subclones. And then with all of the correlative data combined, we expect to see resistance patterns varied within a single patient, and we expect also to see that these lesions respond differently to different TKIs. We're open and enrolling at 4 sites, shown here. And so far, the enrollment is on track. We've almost completed the main study, and then in the PET-CT cohort, that one has been slow primarily due to the logistics of identifying the patients with biopsyable disease and was only open at Dana-Farber initially, but we've now finalized contracts and shifting funds to allow this cohort to open at all of the sites. So I expect the last bit of enrollment to go more swiftly and expect to have results in the summer of 2027. So with that, I want to thank all of the participants, all of the sites, the investigators, SARC especially for all the support, for bringing us all here together today, and then lastly to our funders as well. And I know time is limited, so I'm happy to talk more at our poster session on Saturday morning. Thank you.
Thank you, Candace. We are now going to present 2 studies that we are planning to activate in 2025. The first one will be presented by Dr. Napolitano from SARC-45.
Good morning, everyone, and thank you very much for giving me the opportunity to present this very exciting and ambitious trial of phase 2 trial of tebentafusp in HLA-A0201 positive patients with advanced clear cell sarcoma. This is a study that we jointly developed with Memorial Sloan Kettering Cancer Center, in particular with Evan, who unfortunately is not here today. And I'm happy to present on behalf of both of us. As you well know, clear cell sarcoma is an ultra-rare sarcoma with a very limited therapeutic benefit from available systemic treatment. And so it's one of those cases where we really need to try to come up with new treatments and thinking a little bit outside the box. And one of the things that we notice, typically one of the diagnostic markers of clear cell sarcoma is a melanocytic marker, which is called HMB45 when you do the immunohistochemistry. But if you look at what is the antigen that HMB45 really is, that's actually GP100, which is an immunogenic melanocytic peptide that's been well known and well studied in the melanoma world for many, many years. And very recently, tebentafusp has been approved in uveal melanoma for specifically targeting this antigen, and this is the first FDA-approved bispecific T-cell engager with a CD3 binding effector end and a targeting hand that is basically a TCR recognizing the HLA-O2 bound to a GP100-derived peptide. So both Evan and I came up with the idea, okay, why don't we try tebentafusp in clear cell sarcoma? And this is how the project started. And the primary objective is to estimate the proportion of HLA-positive patients with metastatic unresectable clear cell sarcoma treated with tebentafusp who are progression-free at 24 weeks. And we know from the melanoma study that objective response is not the best radiologic marker of efficacy for tebentafusp, and that's why we opted for progression-free 24 weeks. And there are secondary objectives, the usual ones. Of course, the study will be accruing patients with an ultra-rare sarcoma and the right HLA. So we're really looking at very, very selected population. But to try to improve the relevance of the study, we also decided to include an observational cohort of patients which are the HLA negative, the patients that would be otherwise called screen failure. And this will be treated with physician choice, which can be chemotherapy, TKIs, or other treatments. And the idea is, even though this is not a truly randomized study, because the 2 cohorts will differ is on the HLA, at least we're gathering prospective data on a cohort of patients, which, if we assume that the HLA is not prognostic, may be sort of used as a randomized control arm. Eligibility criteria is the usual, so we need to have histological confirmation of HMB45 positive clear cell sarcoma with the right HLA and measurable disease, adequate organ function. This is how the tebentafusp will be administered, and this is derived from the melanoma study. We know that there is a relatively high risk of cytokine release syndrome, so the drug is given with escalating doses. The first cycle is a lower dose, and then the treatment is increased up to the full dose of 68 micrograms. And as I was mentioning, we have a screening phase. HLA-positive patients will receive open-label single-stage tebentafusp, but we will also collect data on the HLA-negative cohort treated with physician choice. At the moment, there are 3 sites which have confirmed participation. We're still waiting for the 4th site, and I've been told that we will know more by the end of this week, and hopefully we'll be able to start later this year. Thank you very much.
Thank you. The next study will be presented by Dr. Michael Wagner, SARC-46.
All right, thank you. So this is just very newly minted SARC-46, and I think for the sake of time, I'll try and go quickly. So this is a study for EHE, epithelioid hemangioendothelioma, which, as you all know, is characterized by sort of classic fusion proteins. There are only about 100 cases of EHE per year. There is some rationale for using mTOR inhibitors in EHE, and from this really initial preclinical data, actually a number of sites and a number of sarcoma oncologists are using off-label sirolimus, and some of that data has been studied, primarily or really only in a retrospective fashion. The largest of these data sets has 38 patients from Italy, where the median PFS with sirolimus was 13 months, which is a bit more favorable than a lot of the other systemic therapies that we use for patients who have EHE. And the response rate was about 10%. This is just an example of a patient who responded. This study, SARC-46, will use nab-sirolimus, which of course is albumin-bound sirolimus, and has some potential benefits over just the older sirolimus. As you know, this is FDA approved for PCOMA, and importantly, in the studies that led to that approval, there were patients who had previously progressed on sirolimus but then had benefit from nab-sirolimus. The SARC-46 study is a single-arm phase 2 study that's not randomized, so all patients will get nab-sirolimus, and it's a 2-stage design where the first stage is 12 patients. If there's 1 response or more, that will open up a second stage of an additional 25 patients for a total of 37 evaluable patients. The primary objective, as you probably gathered from the last slide, is the response rate. We will, of course, look at the usual secondary and exploratory clinical outcomes, as well as quality of life, where at least as it's currently written, it incorporates the EQ5D3L PRO. Of course, this is not statistically powered for those endpoints, and then we've also included a number of correlative studies to look at the specific fusions that patients might have, and for phosphorylated S6, which is a biomarker for sirolimus and nab-sirolimus responsiveness. And then finally, we'll look at MYC1 levels in the blood, which have been previously correlated with response in EHE specifically. I'll sort of skip that slide. Key points in eligibility. So as the title states, this is for patients with progressing or symptomatic EHE. So we're not requiring radiographic progression, and this is to a certain extent up to investigator discretion. Some planned subgroup analyses are the analyses by fusion type, and importantly, the presence or absence of serosal effusions going into this study. In our grant submission, we had to pre-select a number of sites. So the 5 sites that you see are the 5 sites where we're at least initially planning to open. So thank you to all the investigators who helped with our grant submission. And along those lines, we were able to secure a grant from the DoD to fully fund this trial. So thank you.
Thank you. Thank you all. Thank you, the PIs, for the ongoing studies that we do have, and more importantly, the active studies that we're going to activate in 2025. With that, we will then go to the next topic, which would be the Sarcoma Centers Directory. Now, the Sarcoma Centers Directory is led by Dr. Chappie Conrad. He has worked tirelessly along with our leaders at SARC to develop a more patient-friendly, patient-facing aspect of our website. That includes ensuring that the centers that are highlighted are validated. Number 2 is to make sure that the content that we provide is going to be helpful and informational. And with that, we'll have Dr. Conrad kind of share what he has said. So we'll skip these 2, and we'll go to Dr. Conrad on the base of time. Thanks.
We have the slides for SCD directory. It's a pleasure to present an update on the Sarcoma Center Directory, previously originating from a program that's existed since 2003, which was just the centers, SARC Centers map, and has been expanded significantly to include patient education and triage functions. The project initially began with recredentialing the old map, with reevaluating our programs in terms of activity and continued involvement. They were assessed for multidisciplinary criteria, new patient volumes, which was information requested directly from a questionnaire, and also asked for any subtype data which they could provide, which had a very, very limited response rate. In the final analysis with the recredentialing program, which occurred throughout 2024 and continues into the future, but the essential recredentialing of previous programs ended at the end of the calendar year 2024 with 80 North American programs and 20 international programs. There was some confusion over what programs were pediatric and which were adult and which were overlapping, and that was cleared up. And again, a voluntary questionnaire. And the map now that exists online, which will be released here in the next month, contains a teardrop for sarcoma centers, and that teardrop contains a summary page and context for each clinic. There was a process of assessing volumes and programs and making a decision that high-volume programs deserved recognition, so high-volume programs have a highlighted teardrop in the new edition of the program. There was so much interest in subtypes that I couldn't resist the opportunity to start a couple of work groups on subtypes. We have a subtype project beginning within our membership in North America and internationally, where they basically outshine the Americans with regards to prospective tumor registries that are institutionally based. There was a pathology project that I initiated last year with the help of Dr. Joe Laniers at Boston Gene, which met for the 3rd time yesterday, trying to reach some consensus on the diagnostic criteria for UPS, apropos to the discussions of this morning. We ended up with 5 of really excellent pathologists: John Gross from Hopkins, Greg Travell from Stanford, Al Rosenberg from Miami, and John Chisholm from WashU, and a very stimulating addition at the end, who's a fantastic young pathologist, Rex Hung from Dana-Farber Harvard. There's another program that's using AI for pathology reports, in addition to the American and international programs trying to correct subtype data, and Gina D'Amato has helped me sort of manage some of those programs. We desperately need your subtype information if you have it. Because we were into sort of qualifying and assessing programs and into so many different things with the directory, Stephen and I, with Scott's support, decided to establish a steering committee. Steering committee includes our very valuable patient advocacy groups with Annie and Mitch Anchi, Jeff Kramer from Sarcoma, and my medical partners in that steering committee is Seth Pollock, Angela Hung from Sydney, who could not be here today, Nita Samaya, and Bree Wilkie, and they have been great partners for us in discussing multiple issues. Here's a model of the new sarcoma directory program. It's based on a patient education module, which we refer to as the reference guide there, and 3 different triage mechanisms, search engines associated, originating both within the reference document and on their own throughout the website, can easily access those search engines by as you'll see with one of the examples. Here's a homepage representation of SARC's website. In the upper right corner is a menu tab. That menu tab will take you to patient resources, and that patient resource tab will drop down and show you all of the programs listed. I've highlighted them with my expert red penmanship there. Looks like a kindergarten child got loose on the website. Number 1 is the Sarcoma Center map and a listing and access to triage to sarcoma centers, some of which are diagnosis specific, as generated by particular search engines. The reference guide is the reference guide. It's a modification of the WHO. There is a helpline backup. Patient advocacy is a new directory that I created with my valuable assistants, Claudia and Kim. It has 110 patient advocacy groups, and we're sort of favoring diagnosis-specific groups for triage with diagnosis-specific patients. And the last is clinical trials. Here's an example again of the map as it exists now, with some new programs and continued recruitment in the future, particularly with the international group, where we have a particular interest in developing educational programs to assist some of the smaller international programs, especially in Asia. The controversial part of the program is the clinical guidelines discussed in the reference guide. The reference guide is, believe it or not, a modification of the WHO classification for soft tissue and bone tumors, published in 2020. It's been skeletonized down so patients can understand into 10 diagnostic chapters, which contain 70 different soft tissue sarcomas. We will be adding probably the other additional rare diagnoses that are not in this first edition, as documented in the white consensus paper from CTOS, 82 total diagnoses, and rare and ultra rare. Diagnostic alternatives are mentioned. Triage to sarcoma clinics are in each chapter. Triage to patient advocacies are in each chapter, and triage to current trials are in each chapter. The controversial part of the reference guide are the clinical guidelines and some indications with timelines, which will be carefully reevaluated and scrutinized over the next month. Here's what started much of the issue on subtypes, which was our desmoid colleagues at both the Desmoid Foundation and also through SpringWorks, with the program that we had begun with SpringWorks, valuable support, and we very much appreciate their support and their stimulation and frustration with our inability to collect subtype database information for SARC. I think this new program will facilitate much of those initial goals. Active clinical trials is a partnership we initiated with Antidote. We're sort of halfway or 2/3 of the way into that. It'll be finished in the next couple of months. You simply put your diagnosis in the window and it takes you to active trials. And both, I will say, as a general rule, and that my steering committee strongly supports the inclusion of trials outside of SARC, not just SARC trials, but all sarcoma trials, in addition to SARC clinics. So we're going to be adding clinics that are currently not participating actively in SARC, and we'll be politely recruiting them to see if they have an interest in being listed in our directory program. There is a 24-hour, believe it or not, helpline. Guess who is going to be manning that in addition to my buddies in the office? And we are doing that to support questions about both the directory and patient needs for whatever triage they need to have for their disease. So please send me contact me if you have any interest in participating in our subtype database for the future. Send me especially your comments when we make this document and programs all live in the next 4 to 6 weeks. I would love to recruit a couple of new partners to be on my editorial board, because I expect this to be a bit of a living document and to be annotated with appropriate references in the near future. I finished reviewing the bone section, which has not been done yet by this process, and it's going to be work in the future if this document leads to what we expect it to lead to. Thank you very much to the steering committee partners, especially our patient advocacy partners, our sponsorship partners for this meeting and every other part of SARC, and especially to Stephen and Claudia and Kim for their incredible work effort over the last 6 to 7 months. Thank you very much.
Thank you, Chappie. We are running just on time for all of our sake, but we're actually running behind. Stephen?
Does anybody object to if we skip the break so we can make up some time? If you have to use the restroom, please do so. We won't watch you. Bottom line is, I think if anybody object to proceeding, keeping the agenda.